DNA replication. DNA replication is the fundamental process by which a cell duplicates its genetic material, ensuring that the genetic information is faithfully transmitted to new cells during cell division. This process is essential for growth, development, and the perpetuation of life. Key players in DNA replication: DNA helicase, single-strand binding proteins, SSBs, primase, DNA polymerase, DNA ligase, topoisomerases. DNA helicase, enzyme responsible for unwinding the double-stranded DNA molecule at the replication fork by breaking hydrogen bonds between complementary nucleotides. Single-strand binding proteins, SSBs, proteins that attach to single-stranded DNA regions exposed during replication, preventing them from reannealing and protecting them from degradation. Primase, enzyme that synthesizes short RNA primers complementary to the DNA template, providing a starting point for DNA synthesis. DNA polymerase, enzymes that catalyze the synthesis of new DNA strands. They add nucleotides to the growing DNA chain, ensuring correct base pairing. DNA ligase, enzyme responsible for joining the Okazaki fragments on the lagging strand and sealing any nicks in the backbone. Topoisomerases, enzymes that relieve the tension generated by unwinding DNA by creating temporary breaks in the DNA strands, allowing them to rotate. Steps of DNA replication are: 1. Initiation. 2. Unwinding and formation of replication fork. 3. Priming. 4. DNA synthesis. 5. Leading strand synthesis. 6. Lagging strand synthesis. 7. RNA primer removal and fragment joining. 8. Proofreading and repair. 9. Termination. Initiation. DNA replication begins at specific sites called origins of replication. Initiator proteins recognize these origins and initiate the unwinding of the DNA double helix. Unwinding and formation of replication fork. DNA helicase unwinds the DNA by breaking hydrogen bonds between base pairs. This creates two single-stranded templates for DNA synthesis, forming a replication fork. Priming. Primase synthesizes short RNA primer complementary to the single-stranded DNA templates. DNA synthesis. DNA polymerases add nucleotides to the growing DNA strands, using the RNA primers as starting points. DNA synthesis occurs in the 5' to 3-direction. New nucleotides are added to the 3-end. Leading strand synthesis. The leading strand is synthesized continuously in the same direction as the replication fork movement. Lagging strand synthesis. The lagging strand is synthesized discontinuously as Okazaki fragments, each starting with an RNA primer. RNA primer removal and fragment joining. RNA primers are removed by exonucleases, and DNA polymerases replace them with DNA nucleotides. DNA ligase then joins adjacent fragments, creating a continuous strand. Proofreading and repair. DNA polymerases have proofreading activity to correct errors during DNA synthesis. Mismatch repair mechanisms further enhance accuracy. Termination. Replication forks meet at termination sites, and specific termination mechanisms halt replication. In summary, DNA replication is a complex and tightly regulated process. It ensures the accurate duplication of genetic information, which is crucial for maintaining genetic stability and passing on genetic information to future generations.